I don't know about the rest of you, but for me, one of the exciting parts of coming to a workshop symposium like this is it's a chance to really see the impressive gains that have been made on so many different fronts. And in coming to this and thinking about this symposium and workshop, I tried to think what I could contribute to it. And this is a section I know on kind of long-term monitoring, and there's some aspects of that I want to talk about. But the part that really struck me in reflecting back on the 35 years of research I've been involved with isn't just the development of the new techniques that have been really exciting to see and that we've heard about, but something that's come with them, and that come with those new developments, which is the increased collaboration among researchers. And I've realized that really goes hand in hand with some of these new techniques, because when I first started out, we had a philosophy: you could learn anything in a week. And I'm not that arrogant anymore, and I'm older and I can't learn things as quickly. And things have gotten much more complicated that isn't even possible, even if I could. And I've learned it's a lot easier if I am involved in studies and we want to add a component than trying to cram and learn how to do it. You can contact your fellow researcher who is far more capable of taking on that technique than you are and work with them on that. And that's a lot more pleasing way to go. But thinking about these collaborations, it made me think a little bit about the fact there are first of all, a number of reasons why we should collaborate. It's often impossible to take on certain studies, and I'm going to give you some examples, as a single group. There's often a mix of complex strategies and approaches that you can't be an expert on. It often requires kind of a broader geographic base that requires collaboration in multiple areas. And also, I think something that's important to consider is that when you involve many people, it also is a good way to help bring about both kind of political, scientific support for certain conservation measures. And having that sort of broad involvement will often also result in a better and more successful application of the research results. So in thinking about collaboration, I wanted to look at three examples, and I kind of put these into three categories as I thought about collaborations. SPLASH was one I wanted to refer to, and it represented what I consider the broad geographic collaboration. And I'll talk about how SPLASH wouldn't have been possible without a large number of contributors. But largely it relied on a large number of contributors doing similar things in many different areas. The second kind of collaboration I want to give an example of is what I'll call something that's multidisciplinary, which is combining very different skills to conduct a complex study or design. Maybe in one particular area, maybe even all of you packed onto one boat, as the example I'll talk about involved. But people bringing very different levels of expertise, and again, doing something that you couldn't do otherwise. And then the third one is kind of what I'll call comparative, where different methods often try to answer the same question are used, and by comparing the outcome you can gain insights you wouldn't have gained from any one approach. So starting out with SPLASH, SPLASH was really aimed at some very basic objectives, looking at abundance and trends of humpbacks, population structure, and some human-caused injuries. And we think it was one of the largest collaborations. You guys in the Southern Hemisphere may be coming to rival that. We'll see the direction that some of these projects go. But there were about 400 participants in SPLASH from a little over 50 organizations and 10 countries through the North Pacific. And again, the technique was, the field techniques were pretty basic. They really involved collecting photo IDs and collecting samples. What was done with those often was more sophisticated, and I think many of the approaches we heard about in terms of the molecular techniques, for example, have been applied to some of the SPLASH samples, including some of the genetics work, stable isotope analysis, contaminant analysis have all come out of those. But the basic field technique that most of the collaborators were involved in, its hallmark, was using a fairly basic technique, but applying it over geographic scales that went beyond what we could pull off individually. And this just shows the North Pacific, and let's see if I can, yes. And you can see all of these little boxes show areas where there was effort, and this allowed a more complex picture of things like movements of animals among areas, and those, in this case, these lines connect where photo-identified animals were identified in multiple areas, so lines connect resightings of the same individual. Now, how SPLASH was run was basically with this broad number of collaborators, which largely were the researchers already engaged in research in a particular area. And then with a steering committee that was made up of both some of the funding agencies, but also representatives from each of those regions made up the steering committee. And there was also some effort to bring in some of the different disciplines and expertise that were involved in that. So I think that steering committee, by, again, involving the people in the different areas, was an effective way to kind of maintain that level of involvement. But another key part of SPLASH was that it also recognized a certain degree of autonomy in those different research groups. So while data was being pooled and examined at a central location for photo IDs and samples were being analyzed, the collectors in each region could retain access to their samples to use them in their own studies, retain access to their photo IDs so they could become part of the long-term studies in each region. So in the U.S. West Coast, where I do a lot of our work tracking humpback whales going back to the mid-1980s, we were able to maintain that long-term data set while still participating in this broader geographic study. And I think that's a key element of selling a collaborative project to different researchers. And at the very end, I'll talk a little bit about some of the kind of pitfalls, but a key element that you want to represent is that, number one, you're able to achieve something collaborating that you couldn't achieve independently, but also that none of this impinges on your ability to use your research and your results in the same way you would have if you were working independently. So it's only an add-on and not a sacrifice. And now the second example I want to give is one that, and I've drawn a slide here from a generic slide that actually came from an early study looking at the effects of low-frequency active Navy sonar on marine mammals. And there have been a number of these. This was one of the earlier ones, looking at behavioral response of animals to playback of different anthropogenic sounds that there's concern with. And in this case here, I'd like this diagram, and I'll just point out a few things. You might have a sound source. In this case, it was a ship producing it, but it could be a sound source deployed to do playback of a simulated sound. You have observational platforms that can do behavior monitoring, small boat operations that can do photo ID and deploy tags to get dive data. In this case, there was an aerial component. So you have a variety of these techniques being able to be used. And the more specific study I want to talk about is the one that started in 2010 called the SoCal Behavioral Response Study. And this is only in its second year of a 5-year study, but still it's worked out pretty well. You can get a little bit of a sense of the collaboration by all of these logos at the bottom that are trying to squeeze in all the different groups that are participating in this study. And just to look at a few components of that, for example, in the case, and I'm not going to focus on results of this, but again talk about the different pieces of this and what it involved. We did have a central platform that served as a base of operations, and we managed to crowd, I think at its peak point, about 17 different researchers that represented different components of the study that were engaged, that include visual observers that would actually play a role in spotting and tracking animals. There were photo identification obtained, and that often linked to long-term catalogs in the case of, for example, blue whales, that was one of the key study species, but also some of the rarer species like beaked whales. There was passive acoustics on 2 different bases. One, a array that the Navy operates west of one of their key base of operations, San Clemente in Southern California, but also increasing use of a towed array, passive acoustic array, to track animals as well. There were small boat operations to deploy tags, and in a lot of ways the small boat operations also were the ones going out, finding the animals initially, identifying animals to tag, deploying the tags, and then the small boats took the lead role in doing some of the tracking and behavioral monitoring of the animals once they were tagged. There was a geographic information system to try to show all of this in a real-time way so that the playback of sound could best be set up in terms of location and timing to deal with the target animal. And then there also required some development of a custom sound source that could be small and rapidly deployed to play back, in this case, these mid-frequency sonar sounds. So all of that involved different personnel on this team to do this, and it turned out to be a very successful operation. In the first 2 years, quite a number of animals were able to be tagged with these suction cup tags to note behavior. There were 46 of these controlled exposure experiments conducted, so a wide range of species and playbacks, more than we hoped with this kind of an operation. And if you were, with all of these different approaches being used, that often complemented what you were getting on a particular species. It was potentially to the detriment if you were the only member of a particular species to be in the area. So sperm whales were one of our target animals, but there was only one adult male sperm whale in the Southern California area. And photo ID helped show us that this was actually a regularly returning animal over a 15-year period. And here you'll see, here's a Mark 10 GPS tag to get position. Here's a acoustic D-tag on this animal. Here's one of Russ Andrews's limpet satellite tags. So the animal was well instrumented. It gave us everything from dive data, acoustic record of here's the playback of the mid-frequency sound overlaid with the clicks of the sperm whale to look at how these received sounds might alter its behavior. We were able to get very detailed movement data on the animal in the Southern California area as it worked back and forth several areas of the shelf edge, as well as long-range movement down towards the Galapagos Island with the satellite tag. So all of these kind of played together to get a very clear picture of what this one animal was doing. We're also in a much more challenging species to work with, the Cuvier's beaked whale. We've been able to get 2 more of these deployments and playbacks on the species that was the species of greatest concern because of evidence of high sensitivity to mid-frequency sonar, but represented a very challenging species to study. But I won't go into detail on that, but just to say that this collaborative approach turned out to be very successful. And now the third kind of comparison I want to talk about is where a similar, where you use a comparative approach, and in this case we have long-term studies of humpback and blue whales with photo ID. They've given us very nice trend data in the terms of how things have changed over time off California. This is the abundance of humpback whales over many years and how that changed. But it also allowed us to compare to the trends we were seeing with blue whales. And because of that difference in what we were seeing in terms of trends between humpback and blue whales, it raised some questions about what was going on with blue whales. And I'm going to leave the rest of the story to largely be told by Jay, which is going to be comparing some of the different approaches like line transect and mark recapture, because this was a case where the use of 2 different approaches that get at abundance but different aspects of abundance ended up revealing something about what was going on with blue whales that neither approach would have told us individually. We would have come away with 2 different conclusions, neither of them telling the complete picture from using only one of those techniques. And I hope Jay will have a chance to kind of explain that a little more. So mostly, how do we encourage collaboration? You know, again, that point of emphasizing it's an add-on, and any research group that's invested a lot of time and effort into their research is going to be concerned in a collaboration of, are they giving something up? Are they losing access to their data? Are they going to lose credit or acknowledgement for the work that they do? And it's really important to kind of acknowledge and address those, because to have a collaboration, it really requires that you figure out a way to address those concerns and fears. And it's important to allow some level of autonomy in these individual groups and provide a mechanism for group control and management, that they feel they're a part of that collaboration. It's also important to achieve what I'll call critical mass, because at a certain level, especially in broad collaborations involving groups engaged in the same thing where there's a natural element of competition, once you have a core group that's enough groups involved with it, that really does encourage everyone to be a part of it. It's also important to address those challenges. That includes publishing rights, recognizing how different groups are going to get credit, and preserving those contributors groups to their access to the data and samples. I think those are all important elements. So the very final point I really want to make is, if all of these techniques excite you and the idea of collaborating excite you, where do you begin or where do you start? And that's where I want to point out one of the most critical ways that collaboration begins is at symposium and conferences like this. So if you're here and you're interested and you're looking for how to continue your research, these next few days are really the chance to set up arrangements with people. If someone's using a technique you think can be valuable for you to work with, this is the time to talk to them. Take advantage of these opportunities. It's the main reason I think many of us enjoy coming to these conferences. We recognize it's that chance to meet and talk to other researchers, but really what we're talking about is it's really the chance to set up collaborations and set up new arrangements that are going to be beneficial to both parties. So I urge you to look for those opportunities here, and I really thank the Barbara and the whole Southern Ocean Research Partnership for holding this, because I think it's the perfect way forward. Okay, thank you.